Hey everybody, thanks so much for joining us from Genes in Space. My name is Mark Bliss. The Genes in Space competition has asked for your questions, and we're here to answer them. I'm joined by Dr. Matthew Smith. Matt's been a part of the Genes in Space community in mentorship roles since 2018. He's mentored 4 teams across various stages of the Genes in Space process. That includes mentoring students on creating and fine-tuning research proposals for Genes in Space, adapting winning experiments for the International Space Station, and also working with students on writing the resultant manuscripts. So Matt, thanks so much for joining us today.
Yeah, thank you for having me. It's an honor to be here.
My first question to you is what is Genes in Space, and what benefits do you think that the program offers to students who are interested in space and in science?
So when I love the Genes in Space program, to me, what it represents is an opportunity to get young scientists, so students in middle school and high schooler that are interested in biology, space, and the intersection of those two, giving them a platform to really begin to explore what those fields look like and find current interesting topics to really dive into and really kind of unleash their creativity in asking really interesting basic questions about biology in space aboard the International Space Station. And, another reason why that's such a really exciting kind of space to get ideas from young students is that there's a lot of open questions about how basic biology, how that changes on the International Space Station. And so I think it's looking to the next generation of scientists to really, who will be kind of leading that generation in how is biology happening in these kind of low resource microgravity influenced environments aboard the ISS.
And you've served as a mentor in previous competition years, including one year where your students won and you helped to work on this project for the ISS. How does planning an experiment for the ISS National Lab differ from experiments on Earth, and what restrictions did you not expect?
Yeah, that's a great question. Some of the biggest differences are, just the tools and the resources available on Earth in these really big research institutions versus what tools and resources are available on the International Space Station. So the first thing we can think about is just the space and the constraints and tools and experiments that you can run on the International Space Station. So if you were looking through the toolkit for Genes in Space, you're probably seeing a lot of smaller, really fantastic tools that can be run in low resource environments like the mini PCR thermocycler or the fluorescence viewer, or even things like biobits, right? These cell-free reactions. So often on Earth, we're using kind of big sequencing machines, like almost the size of refrigerators, that take up a lot of space. So part of the restrictions on the International Space Station is just downsizing everything and making a very efficient research pipeline. Another really interesting difference is thinking of the astronauts that are actually conducting these experiments. So they have incredibly busy schedules. The time that they have to conduct the experiment is pretty restrictive. When I'm designing experiments on Earth, I can redo experiments many times if I make a mistake, mishandling a sample, or if I need to repeat an experiment across another day. If a sample is kind of dropped or mishandled, right, it's a lot more challenging to get replacement samples up to the astronauts, right? So by the time we're sending a protocol to the astronauts on the space station, it needs to be pretty well refined and a really well kind of working and described protocol, rather than what I'm normally doing day to day, which is kind of experimenting around, playing with different methodologies and ways of optimizing a workflow.
So if a student wanted to choose a model organism, how would they go about picking which one might be right for their hypothesis?
I like to do a background kind of literature search. And this also can be, using more modern tools, so science, channels on YouTube or other social media. But looking for what is the current state of research in that field and what organisms have been previously used to study that process. Another thing that I think is useful and relates back to that point I mentioned is that I like to look for the simplest system at which this process can be studied, and your hypothesis can either be confirmed or rejected. Looking potentially first at cell-free environments, microbial communities. Have researchers started to, let's say you're interested in a particular protein, have they started to put this particular gene in bacteria and express this gene and look at protein production? Or are you more interested in a kind of downstream process, more on like cytoskeletal function, or even looking at the function of a particular tissue? Does that mean that you can't work with microbial communities, but now you're looking at more of the multicellular organisms? So moving up, there's another really well-studied organism called C. elegans. This is a type of nematode, still very tiny. You can put them on petri dishes, easy to transport, but you have a bit more function. You can measure a behavior, as well as doing some genetic manipulations, right? And following that logic, right, you could think of even scaling up to, oh, are you interested in, say, processes of osteoporosis? So what are the, if you're interested in actually what's the impact on bone, then are you looking at vertebrate systems? Again, that brings a lot of challenges in terms of how that system can be studied on the ISS. So can you take that vertebrate system and kind of look at ways of measuring that or testing your hypothesis in other organisms? But again, kind of depends on your question. So building up, what are the fundamental things I'm interested in testing, and what's kind of the most basic and well-characterized system that I can get started in manipulating and asking these questions.
So if a student is using the Genes in Space toolkit and they're not really sure which parts of it they should be using for their experimental design, how can they decide which of those tools would be most helpful for them?
Yeah, great question. So the way I start thinking about this question, what type of result are you interested in measuring? And what biological processes do you need to measure to get the result that either confirms or rejects your hypothesis, right? So you can start thinking backwards from there, and then looking at the number of resources on the Genes in Space website to learn more about the tools that are available there. I can list them really quickly and kind of the benefits and use cases of each one. So first, thinking about the mini PCR thermocycler, right? So this is important for polymerase chain reactions, right? This is a fundamental kind of tool within the molecular biology toolkit. Allows you to amplify specific regions of DNA, allows you to, from that, you can then run this on gel electrophoresis and kind of confirm or test the size of a DNA product, right? So if your hypothesis is testing whether the size of there's some change in DNA or potentially the change in a gene product, that could be the tool that you use. Maybe you're interested in gene expression, right? So, and you want to use tools in quantitative PCR. So rather than just looking at an endpoint, which is provided by kind of classic polymerase chain reaction, you can start taking time points after adding in the appropriate fluorescent probes. And so those time points will give you a change in fluorescence, indicating a change in basically the amount of gene product in your sample. And so that would be incorporating the mini PCR P51 fluorescence viewer, right? So that would be the thermocycler plus the fluorescence viewer gives you some metric of gene expression. So now the last tool is the BioBits system, right? So a really fantastic cell-free reaction that allows you to study very specific kind of mechanisms for, let's say, protein production or other kind of complex reactions without a lot of, in a very simplified form. So you can imagine, creating an experiment where, you're interested in, let's say, protein production, And so maybe you design an experiment where you have a fluorescence probe attached to a protein. So you know whenever you have an increase in fluorescence, that means that your protein is also increasing in production. So now pairing that BioBits cell-free reaction with the fluorescence, the P51 fluorescence viewer, now you're getting again that readout over time of how protein production may change in different environments, right? So again, I would certainly refer you back to the Genes in Space website and resources there. But again, I think my strategy is think about the results you expect, start to think backwards. What processes do you need to measure? And then what tools are available within the toolkit to help you measure them.
So I was wondering if you could maybe give some general advice to students on how to pick good experimental controls for their experiments. I know that that's something that a lot of teachers are focused on as these students are working on their experimental designs. How might you go about setting up those controls for an experiment on the ISS?
Yeah, I think that's a great question. And yeah, I think it takes practice, right, to really come up with really good controls. So here's what I think about when I'm designing an experiment and kind of thinking about controls. So first, we can think about the control as almost like a baseline that you're comparing your manipulation to, seeing what if this reaction runs normally versus a variable that I changed, how did these two results differ? So we can think of a very simple example. So let's say we're interested in changes in gene expression aboard the ISS, potentially caused by, let's say, microgravity, right? So the a type of negative control, which would be the negative control being we're going to remove that manipulation that we're interested in from our experiment, would be running that same gene expression experiment in parallel, but on the ground, right, where we don't expect to have influence from microgravity. We're just going to have regular amounts of gravity, right? There are other ways of kind of designing these controls. Another type of control that's really important to think about is a positive control. So that was, I just went over, removing that manipulation effect. A positive control could be, can I recreate what the result I expect to find, but using a kind of standardized, well-controlled method. So if we were to think about that microgravity experiment, a positive control that we can't run in the genes in space environment, but would be on the ground, can we simulate microgravity? So again, adding in that manipulation, but on the ground level, and seeing maybe how that changes your gene expression. Another example maybe we can think about is, let's say that on the International Space Station, maybe there's an effect of temperature, maybe the interaction of temperature and microgravity. So thinking about this experiment, you might be interested in trying to tease apart what is the impact of temperature and what is the impact of microgravity on the results that on my particular experiment. So again, we can come back to Earth and design. We have our negative control, right, of running this experiment without gravity, but maybe you can design a positive control at which on the ground, you're adding in some temperature manipulation that maybe replicates the type of temperature environment you'd expect to see aboard the International Space Station. Again, just showing that, hey, I can recreate this environment in this controlled setting that I expect to see aboard the ISS, and this is the result that I begin to see.
Matt, thanks so much. We have one more question for you, which is, what advice would you give to students who are interested in both biology and space? How might they pursue both?
Yeah, great, fantastic question. Yeah, and this is a time when I think, both biology and space are really exciting and growing fields. And the intersection of those, of course, is really great space where I think a lot of careers are going to be opening up in the future. What I'd suggest, looking even before college, at the middle school, high school level, looking to programs like Genes in Space can really be a great resource to start thinking about, what are the questions that are exciting to you? but also staying current with what is ongoing research and even thinking about how could I potentially get involved. And again, through programs like Genes in Space, that can literally be a reality, which is really amazing. So even if it's just looking at the Genes in Space website, there's a number of kind of education tools, resources about current updated publications through the research happening with Genes in Space that just reading through and being updated with, I think is so important and can be really useful. There's also a number of programs similar to Genes in Space that are run through NASA. And we can list a few of those in provided links. Also, I think a really kind of important part is staying current on research happening between space and biology is finding community of other people that are interested in those topics. And even if it doesn't exist at your school currently, even beginning to create that space where you can start to discuss these questions, maybe talk about some papers, ongoing research, what's happening aboard the ISS, even turning to social media, right? Get creative, create this broad platform, right? It doesn't need to be broad, it could be for yourself, but a way of kind of sharing your interests and passion and giving yourself an outlet, show your knowledge about biology and space and learn. Yeah, so I think those are, kind of the core ways to kind of get started and continue moving through the fields of biology and space. And I think all of that still applies once you get to the college level, right? There's a number of internships through NASA that, again, can help get you actual working experience within that space. NASA also has even career opportunities down the line that they list, but also the point about creating community and sharing your passion about biology and space at the collegiate level, I think is important and can, again, help increase your knowledge about the space and kind of share it with others. So.
On that subject, Dr. Matthew Smith, thank you for sharing your knowledge with us today.
Yeah, thank you. Again, it's been a pleasure.